winner.
The plasma membrane is a movable structure that surrounds every living cell. The plasma membrane controls what molecules enter and leave the cell by selective permeability. This allows the cell to maintain homeostasis, which is a stable internal environment. The plasma membrane consists of a phospholipid bilayer that is made up of a glycerol phosphate group and two fatty acid tails. The glycerol and phosphate group make the hydrophilic head polar, and the fatty acid tails make the hydrophobic region nonpolar. This makes the membrane amphipathic. The plasma membrane also contains proteins, cholesterol, and carbohydrates. Passive transport requires no energy from the cell to transport molecules through the plasma membrane. The first passive transport is diffusion. Diffusion is the movement of molecules from high to low concentration until equilibrium is reached. Equilibrium is reached when water enters and leaves the cell at the same rate. Nonpolar molecules can go directly through the lipid bilayer because the nonpolar region is attracted to these molecules. Polar molecules and charged molecules/ions do not pass through the lipid bilayer easily because the nonpolar region rejects them. Therefore, polar molecules and charged molecules use facilitated diffusion to pass through the plasma membrane. Facilitated diffusion is the diffusion through a transmembrane protein of polar molecules and ions. The four types of facilitated diffusion are osmosis, channel proteins, ion-gated ion channels, and carrier proteins.
Osmosis is the movement of water across a membrane through a protein called aquaporin from a high concentration of water to a low concentration until equilibrium is reached. Tonicity is the ability for a cell to gain or lose water. The first type of tonicity is isotonic, and this is when the water enters and leaves the cell at the same rate. Animal cells will prefer this solution, while plant cells will wilt and lose their turgor pressure. Hypertonic is when there is more water leaving the cell than entering the cell. Plant cells will die and plasmolysis will occur, which is when the cell membrane detaches from the cell wall, and animal cells will shrink. Hypotonic is when water enters the cell at a faster rate than it leaves. This will cause the animal cell to lyse, while the plant cell will prefer this solution because it has turgor pressure, which limits how much water enters. Bulk transport moves molecules across the plasma membrane. This requires energy and can happen with two ways, endocytosis and exocytosis. Endocytosis is the engulfing of large molecules or nutrients to then transport across the membrane. The first type of endocytosis is phagocytosis, which is when the food particle is engulfed. First, the food particle binds to the plasma membrane before it is then completely engulfed into the cell. The second type is pinocytosis, which is when the cell engulfs extracellular fluid for the molecules dissolved in the fluid. Third type is receptor-mediated endocytosis, which is when the cell engulfs a specific molecule called a ligand using a receptor to bind to it. Exocytosis is when the cell secretes waste into extracellular fluid by traveling through the cell.
The second type of facilitated diffusion is channel proteins. Channel proteins provide channels that allow specific molecules or ions to cross the plasma membrane. These ion channels function as ion-gated ion channels that open and close in response to stimulus. The channel opens when one molecule bonds to the channel protein. The lid/gate closes when the molecule is no longer bonded to the protein. The next type of facilitated diffusion is carrier proteins. Carrier proteins undergo a change in shape that changes the solute binding site across the membrane. The molecule enters the top of the protein binding site and is then pushed through the protein out the other side. The protein changes shape as molecules pass through. Active transport requires energy in the form of ATP to move molecules through the membrane from a low concentration to a high concentration. This goes against the concentration gradient by using a protein pump such as the sodium-potassium pump. This pump actively transports sodium out of the cell into the area surrounding the cell. At the same time, potassium is being pumped from the outside into the cell. The hydrolysis of ATP provides the necessary energy for the transport protein to continually change shape. 3 sodium ions are exchanged with 2 potassium ions to maintain homeostasis within the cell.
The end.